Let's put together our DNA structure paper model. I've already printed and folded in half the base pairs. So DNA is made out of a phosphate attached to a ribose sugar or a deoxyribose sugar. That's where the name DNA comes from. It's deoxyribonucleic acid. Now this one has the purines on it, and they are adenine and thymine, and they're held together by 2 hydrogen bonds. The pyrimidines are called guanine and cytosine, and they're held together by 3 hydrogen bonds. Now DNA is put together in what we call a 5' to 3' direction, and that refers to the number of carbons on the deoxyribose sugar. So the phosphate is attached to the 5' sugar, and the base is attached to the 3' sugar. So the way we put this together is we attach the phosphate, which is the 5' end, with a little piece of tape. We're going to attach that to the 3' end of a sugar here and just stick it together. Now, people say DNA is anti-parallel. That's because it's 2 strands put together and they go in the opposite direction. So if we flip over the model, we have the 5' end here, and we can attach it to the 3' sugar. So can you see now how this goes in opposite directions with this bit sticking out on this end, and underneath it's sticking out on that end. If we flip it around, it's the same. So we're going to go ahead and put together the rest of the bases, and then we'll talk a bit more about DNA. Add the base pairs one at a time, and then fold between the phosphate and the sugars. So here is our DNA molecule. It's got a 5' end that goes to a 3' end. It's a double-stranded molecule. It's an anti-parallel double-stranded molecule because the other side starts with a 5' at that end and the 3' is at this end. We read a DNA sequence from the 5' end to the 3' end. So this one is AATTACCCAGGG. DNA is often shown as a ladder, but how do you make that ladder into the double helix that we also see? All you have to do is twist it.